The theme of this edition in El Aleph, organized by UNAM, is related to the possibilities and care of life. And we have two great experts that have delved into the very first expressions of its origins and its threats, such as bacteria and virus. They are the Nobel Prize in Chemistry Ada Yonath and Professor Antonio Lazcano, a distinguished researcher at the Science Faculty of UNAM and member of the National College in Mexico. Welcome. It's a pleasure to chat with you here. Welcome, Ada. Welcome, Antonio Lazcano.
Thank you.
Thank you. Hi, Ada.
Hi.
So good to see you.
And you know, Ada, when I interviewed you at Weizmann Institute some years ago, you told me that Professor Lazcano was with you in Israel some days before, and that you talked about a fascinating discovery, a prebiotic structure in the ribosome that could be the link between the inorganic and the organic expressions. That means between inert matter and life. Let's try to spy onto this talk you had with Tonio on this discovery and the origin of life. Let's talk about what transpired at that occasion, or what your talks are all about regarding the origin of life and these prebiotic structures.
So we thought that actually we met first in Florence in Italy. We thought in my lab, when we saw the structure of ribosome, we thought we see there what could have been the first factory for making proteins, prebiotic ones, before life. And until then, I didn't do anything about origin of life. I did other things. So it was, for me, about 6 years until I believed it myself. And then there was a meeting of the International Origin of Life Society. And I decided that I will, in Florence, and I decided that I will go there to see what people know and how can we tell about ours and collaborate and so on. So I applied to this meeting like a student, like a postdoc, and I asked for showing our results, and they asked me back, do you want oral or poster? So I said oral, and they gave me 12 minutes. And in these 12 minutes, I said what I could. And before I even sat down, Tonio jumped and said, you come to UNAM and tell it the same, and so on and so on. This was the beginning, correct?
It's absolutely correct. Yeah. I have very good memories of that.
So,
you know, immediately.
But this is very, very interesting because it has to do with the transition from the inert matter to life. It's trying to explore the very origins of life.
Yeah, correct.
Yes, and Tonio, you have studied with Alexander Oparin. Let's talk about your perspective, your take on the current cutting-edge research on the unraveling of the origin and evolution of life. It has, like we have talked before in some occasions, some Frankenstein-like qualities.
Well, thanks, Pepe. Thank you so much, Ada. You have made a wonderful description of our first meeting and how what has been a very meaningful friendship for me actually started. When at the last century, when Oparin first proposed his ideas on the origins of life, we had no conception, a precise conception of what the primitive Earth was. And I think that one of the most significant things that he did was to put the origins of life in an evolutionary perspective. Instead of saying that you have a spontaneous generation, he said that, or he proposed that you could actually look at the evolution of subcellular structures when he proposed his theory, nobody had an idea of the nature of the genetic material. Nobody had an idea of what ribosomes are. People had very unsophisticated idea of viruses and so on. But I think he really got the point in insisting that life doesn't depend on a single molecule. One of the advantages of his theory was that it was put in the context of the evolution of the planet. Life is not an isolated property from what we see on the earth, on the one hand, and on the other hand, that we don't depend, there's no such thing as a living molecule. What we have are, life is obviously the manifestation of the properties of sets of molecules of different nature. And as time went by, we were able to come with more refined descriptions of what we have as cells, of what minimal cells are, and what are the subcellular structures. Now, we don't want, certainly, to think that a ribosome appears by itself, as ribosome is complex as the one we have today. We don't want, or we cannot think of a membrane like those we have today appearing just like it by itself in the primitive earth. But now we recognize that you can apply evolutionary theory, evolutionary perspectives to try to make a description of the way these complex structures and properties that you see today actually began by very, very simple interaction in very simple versions, very molecular versions.
And that they have some kind of Frankenstein-like qualities, yes?
Well, Frankenstein-like qualities in the sense that you don't require a mystical kind of energy to life to start, but you can see that you can explain life in terms of the laws of physics, the laws of chemistry, and the laws of their interaction. And we don't understand really how life appeared, but I think we have a good idea on how we should look for the answer.
Yes, and let's talk about your perspective when Ada talked to you about this prebiotic structure.
Well, I
discovered that is key for developing life.
Ada is quite right that I was absolutely fascinated, that I jumped immediately when I listened to her, because the ribosome is one of the most complex structures that you see in living cells. The amount of information, of evolutionary information that you see in cells is quite extraordinary. And one of the most complex structures that you see in cells are precisely ribosomes. If you want to understand how the relationship between proteins and nucleic acid started, you need to look at the ribosome. And what she was doing was cutting away pieces of the ribosome, look at the inner sophisticated structure. And for me, it was clear that there was the right path to follow. I have to say that the very same year that Ada was gracious enough to come to Mexico and give a lecture at UNAM, a few months afterwards, I was waken up by the news that she had won the Nobel Award. So it clearly, her work was on the right direction and was finally acknowledged by the scientific community.
So the ribosome is a clever and also stupid organelle. It's big and complicated, and it has many components. In human beings, over 80 components. In bacteria, almost 60 components. But and it took us 20 years to see how it is built and to understand how it works. And when we understood how it works, we could see that the place where the proteins are being made, the site where the connection between the components that make proteins that are called amino acids, the position where the bonds between them is formed is not very large, and the ribosome doesn't do much there except for providing the right conformation, the right structure for them to make a bond. And this piece, which is less than 4% of the ribosome, is the same in every cell, in bacteria, in human beings, in flowers, in elephants. It's exactly the same in all, from the point of view of structure. When I tried to put structure one on top of the other, I couldn't afterwards distinguish between them. So we understood that this part is above evolution. Evolution didn't change it at all. And because it makes the peptide bonds, which is the bonds that were needed to make the first proteins, to make life, it went together. It makes life and it didn't change. The most primitive is like the most advanced one in this piece. The ribosome changes, the ribosome grows, but not this piece. So this is why we thought it's the center of starting origin of life. And in principle, it can happen in the desert, in hot springs, in cold springs, in another planet, and so on. So when I talked about it and showed it, and I was really shaking to say it, because for me it was, you call it Frankenstein? I'm not sure, but it was something new. And Tonio liked it. I was in heaven.
Oh, and how you reacted, Tonio, when you saw this same pattern running through all the cells, through all the expressions of life?
Well, I have spent a good part of my life looking at sequences and structures of biological molecules. So when you see this extraordinary conservation, it's obvious that it has to be important. And when you see that it's precisely in the core of the ribosome, then obviously you have to recognize that it plays a key role in the synthesis of proteins. And but I think it's also revealing about the nature of science, of scientific research. Ada and I had never met. She comes from Israel, I come from Mexico, and we have a common language. And the common language is not English. The common language is a scientific curiosity and the recognition that there are scientific ways of looking at truths that are common. And even when you have different perspectives about the same phenomenon, this can lead to a discussion that can make you understand in a more precise way what you're looking at. Let me add something about Ada that I'm sure she remembers, but she has not mentioned. The last time she was in Mexico, we knew that she was coming, and then I got an email from her saying, with this, like a small girl that looks at the universe with wide open eyes, full of curiosity, she says, I have some new things to share. Would you be willing to let me speak with your students and you in your lab? So, I mean, it's not humbleness, it's this willingness, this intellectual generosity. So we organized a small seminar in the lab. My students could not believe it. I remember they washed themselves, they combed their hair, they put new clothes, they got to prepare coffee, tea, and pastries, and so on. And we have an extraordinary seminar in which Ada presented her new results, her new ideas, and we came out with a possible joint project that has now been stopped because of the current health emergency, right? But that's a way that you see science working. You see someone who has discovered something and is in a very generous way willing to share it with the best people you can have, which are students.
Oh, that's beautiful. And you know, Ada, science is also a story of persistence, because when you see an angle that you try to delve with, you really persist. There is a very interesting story when everybody thought that the ribosome couldn't be seen, but just in computers, that you insisted, and then you had an accident that was a fortunate accident. Could you tell us about it, please?
The accident was with bicycle. I went by bicycle and I fell down right in the beginning of my trip, maybe 100, 200 meters after I started, and got a brain concussion, which was very serious. At that time, about 6 weeks after the accident, I had planned to have a experiment in Berlin, trying to find out, to prepare enough material to understand how the ribosome starts. So each ribosome, each cell has factors that are called initiation factors, and they tell the ribosome to start. The ribosome cannot decide when it wants to make proteins. It has to get a call from the cell. And actually, the ribosome is made of 2 parts. This call makes the 2 parts come together in order to start. So one part is the head, where it reads the code, and the other part makes the protein. So we wanted to make, it was before I went into ribosomes. I thought it's too complicated. So I will look at the initiation factors. But I couldn't come to this experiment because I had this brain concussion and 2 operations, 2 surgeries. So I came about 10 months after it happened, or 11 months after it happened. And they have decided that I'm dead, because when I wrote to them that I will come, it was February, and I became only okay in October. So they decided I'm dead, and they started the experiment. But I came with a student, and the student was there for about 2 weeks. And I stayed my 8 weeks, which I promised them. And it was very good for me, because at that time, first of all, I could find out that they have a lot of good material that maybe I can start with ribosomes. And also, I was very, very weak. So I saved hours and hours in the library just to go on. And I don't read German. I found a journal, tourist journal, that talked about a delegation that went to the North Pole to see how the bears are, what is the way the bears are alive when they are asleep. In my opinion, people couldn't work on ribosomes because they were very unstable. Because when I prepared ribosomes, more than 2/3 were not active. And I thought that I'm not a good scientist. So I wrote to the people that suggested the procedures, and they told me they also get very little activity, less than half of the expected activity. So this gave me the feeling that the ribosomes are not stable, and therefore their structure cannot be determined. But the bears, it was found that the ribosomes are packed periodically in the inner side of their cells when they were asleep. And I thought,
they told this in Mexico, in Spanish. This is sueños de invierno. It's not hibernation, but dreams, winter dreams of the bears.
Yeah, when the bears get up, they need proteins. So they need the factory to make the proteins. And in this packing, they kept them from being destroyed, because they are not stable otherwise. So this gave me the feeling that under pressure, ribosomes can be packed periodically, and this is what we needed for the structure, for doing the structure by crystallography. So I looked for ribosomes from bacteria that live under pressure conditions, like in the Dead Sea, when it's very hot and very, very salty. This was the beginning.
Yeah, that's incredible, because it was against everything that was taught about researching the ribosomes. They thought that this was a crazy idea, that you would persist in this line of research.
That's correct.
Yes.
Okay, so I was crazy. Who cared? I was crazy. I was dreaming. I was out of my mind. There were lots of ways to describe me. I don't know what I did. We had to change many of, or to invent new procedures. It was not going fast and not going smoothly. I must say, there were good reasons to say that I don't know what I'm doing, or that I'm slow, but the worst thing that was what was said about me is that I'm a liar. How come I could do what other people could not? So I'm lying. So this I didn't really like. But in a meeting, I think 15 years after we started, a professor from Yale he wasn't very friendly, but in this meeting he was even less friendly. he tried to avoid me. And at the end I caught him and I said, you know, we heard that you're working on ribosomes now, so what are you doing? Maybe we can collaborate or divide. And he said, collaborate with you? Was not. But he said the second sentence that he said, you know, we repeated your procedure and we got the crystals, we got the results that you reported. So I almost kissed him. Here is my biggest competitor, but he said that I'm not a liar, that he could repeat my experiments. So this was my the minute that I became not a liar.
You know, this has to do with insight and persistence that are essential to science. And while talking about the evolution of life in its very first stages, when we see the evolution of virus at the level of DNA and RNA, Tonio, there is a question that you ask your students that is very important. Do viruses have life or not? What is your answer? And what is the meaning of virus from the evolutionary biology perspective?
Well, I have to say that one of the reasons why I have always been attracted to viruses, although I didn't start to work with their sequences until recently, is because if you are interested in the origins of life, you have to try to understand when you go from something that is not living to something that is living. So you need at least an implicit way of recognizing the differences between the two states. It's not so different when you think about, for instance, issue like abortion. A cell that has been fertilized is alive, but it's not a person. So clearly you have a difference between person and being alive. If you talk about extraterrestrial life, you need to find criteria to define how will you recognize an entity in other planet that is alive, and how do you distinguish purely chemical reactions from, let us say, something that is truly alive. If you think about viruses, you are precisely in the same situation. They do exhibit some properties of living systems, like they can multiply, but only inside cells on the one hand, and on the other hand, they can evolve by Darwinian mechanisms. They mutate, and in mutating, they can acquire immunity against some medications, or they can make a jump from one species to the other. I think that the problem with viruses is that we tend to look at them with a tremendous prejudice that is tainted by their very name. Virus means poison in Latin. And the people think that they are evil, that they are damaging cells. That's not exactly true. And I can quote rapidly 3 examples that this is not the case. For instance, if you look at viruses from the perspectives of bacteria, of prokaryotes, viruses are extremely significant because they are part of the mobility of genetic material that can carry genes that make a bacteria resistance to a given antibiotic from one cell to another. And this is clearly an evolutionary advantage. If you look at the origin of mitochondria, which are the organelles inside cells like those of us that actually perform the biochemical processes of respiration, well, clearly the polymerases that have the mitochondria do not correspond with those of bacteria. Clearly, the mitochondria were already infected by viruses when they became part of the eukaryotic cell. And finally, you have the placenta. The placenta is essential in mammals like us. And when you look at the genes that define the placenta, those genes are homologous, have the same origin as some genes that you find, for instance, in the retroviruses. So I think one should have a larger perspective on viruses that goes beyond the anthropocentric idea as something that is damaging us, look at a wider perspective, and then try to understand how we can cope with them in a way that goes beyond simply in terms of the disease or in terms of the negative effects.
Yes. Ada, what is your thinking on that?
I'm very excited about viruses today because 2 days ago I read a paper that may give some ideas about the question that I always had about viruses. Maybe not always, but many years. So viruses have their genetic information inside them, either as RNA or DNA. But this is less important. What's important is they cannot use it unless they need the machinery to translate this information into proteins, and they don't have it. They are total parasites. This is what they do when they go into cells. They use the machinery of the cell. The most important part is ribosome, but there are also these initiation factors, there are elongation factors, there are termination factors. All these are working together with the ribosome, and there are mobile molecules called tRNA that brings the amino acids, the components. They invade into cells, and they use all this machinery, which is very complicated. How do they do it? So this is a question I always ask myself, how do they do it? Can they recruit all these factors that I just mentioned and some more, and the ribosome, and tell them, now I can do my protein? Can they do this? Do they have enough information to recruit all these components? Or the other possibility, which looked to me more clever, is that they jump into the queue. They find a ribosome that makes proteins with all the factors that is needed around him or will come to him, and they just jump and get out the messenger, the genetic information that the ribosome has, and put theirs. So it's not easy, but at least it's less steps. And I still don't know the answer for this. Maybe Tonio knows, but I doubt. But 2 days ago, maybe you saw it, was a paper in Nature from a big group, part of it from Boston, from the white-headed part, Australian. And they showed, they looked at HIV, not the corona, HIV, but the principles will be the same. And corona is too new. And what they found is that the pieces of RNA, it means the genetic information that the virus brings with him, with the virus, are not just lying in the cell of the host, but each of them has a structure, a functional structure. So when the genetic comes in, the line of the gene of them is being broken to the many genes that they need for their proteins. And these pieces each of them has a structure. It's RNA, it's an RNA virus, has a structure that changes along the cycle of reproducing. So they didn't really answer my question yet, but they gave, for me, they gave a possibility that the virus has more than just invading cells, that the genetic information that it has can also be, it is actually also functional. They decide this, now this has to be translated, now this has to come close to the ribosome. I'm very fascinated by it.
It's very interesting what you're saying. It's like if it has some decision qualities, what is taking place there.
Exactly. So it's not that, of course, it has some decisions to make as an invader. So it will be translated. But
So yes,
how does it convince the ribosome to do his proteins? This is still open question.
Yes, it's an open question about this theme of seeing if virus are in a sort of frontier between life or not life. Yes.
Life without reproduction. But you say, let's think about it like an army coming, conquering a city. So they come in and they use road to the city has the buses, the streets, the shops and so on. Is the virus doing it this way? When it goes into a cell, it has to use all these components, or it can accumulate them, or it just find what it needs, this factor it needs, and makes itself. And if so, how does it convince this factory to take it?
You know, Toño, this is another way of asking the same question about the virus and what it seems to be something akin to intelligence, but we have a question, a frontier question there. So what would be your perspective on this research?
Oh, I completely agree with the way Ada is posing the issue. Let me put an example. You can define a clip, like a clip you use in the office to put, or in your home, to put together some papers. And here you have a very simple definition of a clip. It's a piece of metal, period. I don't know if you can see it. If you can see it, let me put it against my sweater, perhaps. So, or against a piece
Can
of.
I show a picture?
A piece of white paper, like this. No, you're not going to see it. Let me draw very quickly a picture.
I think I have one ready.
Okay, so you can have a piece of wire like the one in the upper drawing, or you can have a clip, a paper clip. It's exactly the same material. It's exactly the same. This is a paper clip. Now you see it.
Yes, we see
okay.
it.
Or you can have the metal here, over here. It's exactly the same thing. It's exactly the same thing. You see the paper clip and you see the wire. But the structure is what's important. So when you look at the way RNA enters the cell, what you are actually looking is not only the piece of wire like this, no, like the piece of wire, but you see that what it's important in biology, it's also the structure, like the clip here. And I think it's the same with the ribosome. It's the same with the RNA in viruses. What counts in biology, it's always the structure, not only the sequences. And precisely when you see the conservation of a structure is when you actually see how you can understand catalysis, control, entrance, invasion, and so on. Now, one thing that I found that it occurred to me listening to Ada is we may not have a good definition of what life is, but clearly, and that becomes very obvious when you think about the viruses. Some people say, yes, it's alive. No, they are not alive, whatever, they replicate, whatever. But clearly, we don't have the same question about the cell. The cell has a ribosome. The cell can synthesize proteins. The viruses, which is the entity about which we are not sure whether it's alive or not, doesn't have a ribosome. So clearly our definition of life requires an understanding of what a ribosome is.
Yes, and let's take another angle that I think that it's very interesting, because when you're studying these finer levels of DNA and RNA, this takes us to possibilities to fight some problems that occur at the very first stages of life. Ada, while doing your research on the finer levels of the cell on ribosomes, you and your colleagues are developing the next generation of eco-friendly specific antibiotics that can eliminate pathogenic bacteria. Let's talk about this approach that helps to preserve life, because this festival, this meeting is about this, seeing also the possibilities of life by having knowledge.
Okay, so I just want to say one more sentence about the previous question of origin of life.
Yes.
Once we thought that we could determine the piece that is fully conserved and can make proteins, we made it in the lab. We repeated it in the lab, and it took us almost 10 years, but we got peptide bonds in this lab. So it shows that it could have been everywhere, and it could have started this way. So it's a piece of beginning of life that could be from some nucleotides that Tonio showed that they can be synthesized under prebiotic conditions, and they came together and they could make bonds. Probably first they did it for themselves, for RNA, but then the amino acids came, and they are quicker and cleverer, so they snatched it and started to use it for themselves to make in the beginning very small pieces, and afterwards, when the ribosome grew and there was already some code and some needs, larger and larger. So this is about the piece of the ribosome that can make bonds. So this piece is almost the same. Actually, it's the same except for its neighborhood in all cells. It means in bacteria and also in human or in animals. And antibiotics that are against bacteria, half of them are working by paralyzing the ribosome. So if the ribosome is paralyzed fully or partially, next generation will have less proteins or damaged proteins. So this is a good way to get rid of bad bacteria, of pathogenic bacteria. So these are antibiotics. About half of the useful antibiotics are made are targeting the ribosome. So when we understood the structure, we determined how every one of these antibiotics is working, and also how resistance to it is being made. And now we understood it all, but we also understood that there is a very little chance to change these antibiotics or modify them so they will be perfect. They can be better, and some companies are doing it. But we looked for other possibilities for antibiotics, and we found them in positions that are far from the main important site, but they are specific. Each pathogen has it in different places. So if we block these positions, if we put something on them, maybe the ribosomes need them, and this way we stop its functionality. And we had 25 sites, we finished with 9, but 9 are good. And this can be blocked by antibiotics that we ourselves plan and design. So we can design them to be not poisoning and easily coming into cells and eco-friendly. So what is important with eco-friendly? The useful antibiotics today, each of them has a piece inside it, a little piece that is not digestible and not biodegradable. So when we take antibiotics, even just the amount that the doctor prescribed, and very, very careful. It's not important. What's important is that these pieces that are not digestible come out, and then they stay in the soil. They stay where the sewage is, and the systems that are doing that are purifying the sewage, the water, the dirty water, don't catch them because these pieces are so small. So they contaminate, and this is known. Near hospitals, the contamination is awful, but not because the hospitals are bad, but because the patients are not digesting these pieces. So when we make our own ones, we don't put pieces like this in them. So it's eco-friendly.
That's beautiful. And you know, this is the result of doing research on the final levels of nature. Antonio, let's talk about your studies on the genomic structure of the virus and the possibilities that could be opened up in terms of developing medicines and vaccines. Because when you know the structure, when you have synchrotrons that are kind of electron particle accelerators that can see into the structure of the virus, when you have the genomic studies of the virus, you can try to develop medicines to inhibit the growth of the virus.
Oh, sure, absolutely. And actually, what we have been doing is exactly the same approach that Ada has been developing. About 5 years ago, we published a paper that was one of the outcomes of the PhD thesis of one of my colleagues. He was at the time working with me for his degree. Now he's a member of the lab. And we found, very much to our surprise, that the way all RNA viruses replicate, the way all RNA viruses multiply, depend on a molecule that I have been in love with for many, many years, which is the polymerase. The polymerase is a protein, a very complicated protein that actually reads the genetic material of the virus and makes copies out of it. And because RNA viruses change and mutate so much, change so much their genetic material, it was not easy to see they were actually coming from the same ancestor. But by now, we have crystal structures. We have the three-dimensional structures of so many different RNA polymerases from viruses that we could actually make a comparison, superimposing the structure exactly in the same way that Ada has described with the catalytic part of the ribosome, the conserved part of the ribosome. And we found and published a paper describing how all of the RNA polymerases of all known RNA viruses, there are so many things we do not know about, had an origin in common. Well, with that understanding, when the coronavirus pandemic started, we decided to superimpose all the RNA polymerase structures to see if they could fit we could feed into them some medication or some inhibitor of its activity. And yes, we found one, which is the sofosbuvir, a medication that has been tried for several years and has been approved by the FDA and other medical authorities, that actually was developed against the hepatitis C virus. And we found that, in fact, it fits so well that it must inhibit the replication of the coronavirus. This is now, the paper has undergone the second review. It's, I think that we will have news about it very, very soon. And it shows how you can actually use an evolutionary approach to try to solve a practical issue, which is exactly what Ada is doing. You make comparison, you describe the molecules, and then you see, okay, I can use this in this way or the other. Now, when our paper was under review for the first time, we were absolutely happy to see that a colleague from Egypt that we have never met so far was using a different approach that is called molecular docking to see if what kind of antiviral molecules could stop the replication of the coronavirus. And he came by different, Gaddafi is the name, Abda Gaddafi. He came exactly to the same conclusion, but different approaches, which I think is precisely the way science works. What I think fascinating about science is the fact that you can always come to the same conclusions, to the same ideas, even if you come from different backgrounds and have different personal or social histories.
Yes, we'll wait a little bit because Ada has disappeared. Voy a lanzar la última pregunta, porque si te diste cuenta, la estructura de todo esto es que hemos visto cómo el estudio de lo que es por curiosidad, el tratar de investigar las estructuras más básicas de la vida, nos lleva tarde que temprano a tener ángulos para combatir bacterias y para combatir virus.
Así es, así es. No, me encanta el enfoque.
¿No? Que eso es lo que estamos planteando y cómo pudimos pasar entonces del estudio del origen de la vida, de sus impulsos más finos y cómo conocerlos nos lleva a esto. Hi, Ada again.
Hi.
Hi.
Do you hear me?
Yes,
Do
yes.
you hear me?
Yes, we can hear you. We are
so
ending
my
this.
computer died and I had to go to my iPhone.
It's okay.
But it's okay. We see you clearly, and this will be the last question we have. I would like to have your thoughts about what we are going through nowadays, and what is your perspective on what is happening in science to tackle this problem of the coronavirus.
So there are a lot of efforts around the world to find a medicine, therapeutics against the virus by intervening with its life, which is with its cycle of life. Like in the beginning, the virus, when it's coming, it has a polyprotein, which is a very long protein that has the proteins that it needs in order to invade and to start its life within the host cell. So this polyprotein is being cut by its own proteins, and they have a way to show that cell that they are better than the cellular protein, somehow that they are preferred. So the idea is to stop these steps, each step at a time. Also, it has, you saw the corona has like spikes, and these are also proteins. So the idea is to find a material that will sit around the spikes and make them less spiky, more like something soft. So then it will not be able to go into the cell. Then these are some effort, but this takes time. There is an idea that the treatments that were made for other viruses, RNA viruses like HIV or Ebola, could be useful also there for corona, because it has they have some common features, and these are being tried today, and even one of them got the FDA approval. Then there is a much more important type of effort, is to make how you call it, to make a vaccine against the virus. And this is also done now. I don't know how long it will take to make it, but this is for vaccination. It means for people that not as therapy. Actually, it's not correct. It sometimes is also good as therapy because it sort of kills the viruses that are in the body of the ill people. I'm sure that there are other ideas too, but these are those that I know about.
Antonio?
Well, if you look at the response from the countries that have managed to cope better with the pandemic, with the illness, Israel is clearly one of them. Germany is another one. Singapore, South Korea, Australia are the other ones. And they have one feature in common. They have invested tremendously in science and in universities. So I think that one has to recognize the role of scientific knowledge in coping with a virus pandemic, with climate change, with a number of other diseases, with the destruction of the environment, and so on. And I think that's a lesson to be learned by all societies. We need to change the way that science had been developing as something that is highly competitive to something that should be highly cooperative. And this requires recognizing that every single society has young men and women as students that can contribute to our understanding of the way nature works and how we can cope with issues that will affect us regardless of the place where we are. So I think that's a lesson to be learned. The other thing that I think is very important is a recognition that we have to make a definition of science that includes other fields of knowledge, like social science, like humanities, and like art. We are now all close up in our homes, and what makes more bearable this situation is the fact that we can read, we can watch wonderful movies, we can visit museums and so on. And that implies that we have to invest not only economically, but also socially. We have to convince society, all societies, that you have to invest in training young people, especially students, into contributing to our understanding, our developing of knowledge. Now, I know this is a dream, but let me go back to what Ada was saying. She was asking how polar bears are dreaming. What I'm saying is, let us have a midnight summer dream, like in Shakespeare, in which we can go into this land of fantasy, in which knowledge can be the common treasure for everyone.
Sonia is completely right. It has to be a common interest of the whole world, and the governments have to support the science. This is really important. It cannot be done just by talking. It has to be done in the lab, and it's not a cheap way of going.
To have this beautiful concert of knowledge and this beautiful concert of literature and art that lifts the spirit and helps to preserve life and the dignity of life. Thank you very much for your participation in Aleph.
Thank you for inviting.
Ada, thank you. Take care, Pepe.
Muchas gracias.
Until next time, take care.
Thank you.
Take care.
Gracias, gracias.
Thank you.